All right. Good morning, everyone, and happy Saturday. Welcome, welcome, welcome. Thank you to those of you who liked the stream even before it started. I think I had one yesterday, and then there were two more. So thank you. That's always encouraging because it means people are looking for it and people are, um, well, they want to hear what I have to say. So I appreciate that. Today I'm going to talk, well, I was going to say we. Today I am going to talk about inhalation toxicology. I'm going to read something that I wrote for the general public. My goal is to get on and off in about an hour. So I'm hoping someone gets something from this and enjoys it. So I'm going to read something that I wrote on this field. And I have pictures that I'm going to show throughout. So again, my goal is to get on and off within an hour, or maybe just over, but not much more than that. So I'm going to play the intro, and then I'm going to start this discussion. If you're here, please shout yourselves out. Let me know that you're there. And yeah, I'll see you on the other side of the intro. I don't have AdSense or Super Chat over here yet. I will get there. But in the meantime, if you're watching this and you want to make a donation, my Cash App and PayPal are here. Those of you who just came in, please smash that like button. I'm going to adjust my camera a little bit so you can see what's on my shirt. This is the original shirt that I wore, I think, in my first offering on this channel. Wow. That was 3 years ago. I started this channel during the World Health Crisis, and I slowly started to build it out during the World Health Crisis. And I was wearing this shirt in my first video, I believe. Wow, time flies by. Anyway, I'll see you on the other side of the intro, and we're going to talk a little bit about, we're going to talk a lot about inhalation toxicology. All right. Hey, good morning, Char Willie. This time I'm looking at the StreamYard side, so I see what's going on in real time. I think in the last stream I was looking at the YouTube side, and I didn't know that there's a lag, and sometimes the YouTube side, at least on this computer, it lags far behind. So it's important for me to keep looking over here. Okay, everyone. Today I'm going to talk about the field and the science of inhalation toxicology. I've covered a lot of things over here, and I've been pondering, discussing this topic for a while. And sometimes it's just time. It's just time to do it. It's time to, and sometimes you just know when it's time. And after my last stream on Henry's Law, that had some inhalation related aspects to it. I said, okay, well, it's time. I I do a lot of these streams on Fridays at lunchtime, but yesterday it was a long week. It was a busy week, and so I had a bunch of other things that I needed to do, and so I just decided to push the stream to this morning. A lot of things going on right now. It's a very busy time. A lot of changes are in motion. Yeah, it's interesting. There are seasons of your life when things are a little more static, and then there are other seasons when things start moving a little bit more. And I'm speaking in generalities here. I'm sure some of you have experienced that as well. Okay, inhalation toxicology. So this is going to be a bit of a well, this is going to be involve science disciplines. It's going to involve um, science careers. It's going to involve, um, yeah, it's going to be a mixture of things, and you'll see as I go through this that it's a mixture of things. And I would encourage with melanated Americans, Black Americans being underrepresented in the STEMs. I would encourage everyone to share this out because there are whole communities of people who don't know anything about this. And there are reasons for that. There was a season before the global health crisis. Look at me speaking in code. There was a season before the global health crisis where I was going and doing career days at numerous schools here in the DMV. And most of those schools were in the Black neighborhoods of Washington, D.C., or in Prince George's County, which are also predominantly Black neighborhoods. And I would go into those schools, and the students and the faculty, the teachers, they would all say a similar thing. They'd say, "You're the first Black scientist that we've ever met." The same thing would happen amongst the federal workforce, the other government workers. The government workforce is vast. There are so many agencies and offices. There are so many program analysts and lawyers and financial specialists and HR specialists, and so many of them that I would meet would say, "Wow, you're the first Black scientist that I ever met." I think that's interesting, but I think it's also telling. By the way, on my original channel, I'm actually going to do a stream. Well, I'm planning to do a stream on not Saturday, on Tuesday night about. Uh, it'll involve looking back over my time as a federal employee. I'm not going to say anything. I don't know if any of my coworkers are watching this, but it's not going to be anything confidential. It's just going to be more general because there are some general things I think that If you are going to work in the federal service, the civilian service, it's a world all its own, and especially if you're new, there are a lot of moving parts and nuances to it. So I have content over there that I created years ago, and people are still watching that content involving working in the federal service. So I'm gonna revisit that. So in my current career in the federal service, I work with a certain class of chemical, which I'm going to keep anonymous for now. But the skill set that got me hired into the federal service was that of inhalation toxicology. And I wrote this back in April, wow, almost 5 years ago. Yeah, almost 5 years ago. Wow. Okay. And I wrote these. I was kind of cutting my teeth. I think that's the expression. I wanted to get started on a potential writing career. And one of the things that I thought was important to write about were the sciences that I was trained in large part because in the communities that I come from, people are just unfamiliar with these things. So this is written for a very, very general audience. And I recently joined the National Association for Science Writers. There's so many worlds and there are so many subgroups and societies in science. Well, one of them is one of them involves science writing. So I was asking this gentleman for advice, and I told him, "Well, most of my background is in regulatory." And he said, "Wow," he said, "Yeah, that's very, very dense, and that's very, very the general public would not want to read that type of writing." And so he said that's less storytelling. That's very, very granular. That's very, very in the weeds. And I realized that the kind of writing that I'm doing now, am I 9 to 5, it's very, very granular. It's very, very in the weeds. It's very, very detailed, and the general public would not understand the majority of it. And so when I wrote this piece here, I did share it with a colleague or two, and one of them who had been in the regulatory world for so long, he did come back to me and say, "Who's the audience for this? Who is this for? What who would read something like this?" Well, there's a difference between writing for a general audience, and there's a difference between writing for a very, very technical audience like that in the regulatory arena. So that's how this piece came about. So let's jump in. Good morning. Oh boy, we got Sheed we here, constructive consultant. Let me shout everybody out because Char Willie, Sheed we here. Thank you for being here. Sheed, I enjoyed that stream yesterday about these prices. The prices are so high. Doug, thank you for being here. And Toya, I think I'll just call you Toya. I'm not sure what they call you these days. Is it Toya Son, Toya the Tutor? Uh, I know it was those two. Anyway, and the couple of times we chatted, Toya has mentioned that she was a grammar snob. So I think I got all the errors out of this piece. So it should be a nice clean read. All right, let's talk about inhalation toxicology. And I'm going to stop at certain points and show some slides. All right. So the quote I pulled, well, this one is called a look at STEM. You guys know that STEM is the acronym for science, technology, engineering, and mathematics. So this is called a look at STEM. What is inhalation toxicology? The quote I lifted from this piece says, while other bodily tissues can tolerate varying levels, while other bodily tissues can tolerate varying degrees of oxygen deprivation, it is well understood that even short periods of deprivation of the brain, oxygen deprivation of the brain, can cause irreversible damage, unlike with long periods of food and water deprivation. I'm sure you all have seen it in the movies. Your brain cannot go very long without oxygen. You can go for a while without food and water, but your brain and most of our bodily tissues cannot go long without oxygen and recover. All right, inhalation toxicology. I haven't read this in a while. With the exception of my blockchain technology post, my previous science, technology, engineering, and mathematics posts have covered the fields of pharmacology, toxicology, and ADME slash drug metabolism, all of which are considered biomedical sciences. Similar to those fields, inhalation toxicology as a discipline dates back to over a century ago and is very complex regarding the wealth and depth of information available. It is also still evolving today. I'm going to talk about that later. As with everything, it's getting more, it's becoming more model rich, more it's moving away from animal testing to more computer modeling and more of a cell-based assay paradigm. So I'll talk about that later. Please smash that like button if you haven't. The goal of this post is not to address every detail and nuance of the field, but instead to give readers unfamiliar with it a basic introductory understanding of discipline. This post was prepared for a general audience, and thus any fellow inhalation toxicologists who may read this may find it a little too simplistic. That's okay though, as the goal is to educate others on the field and what we do. Further details about the many aspects of inhalation toxicology can be accessed online or in scientific journals. This overview of inhalation toxicology definitely falls under my principle of creating ecosystems of success, as it is a very unique knowledge and skill set possessed by only a select few. One of which I acquired accidentally when seeking training in ADME slash drug metabolism as a postdoctoral scientist. Why is inhalation toxicology unique? Why is it a unique skill set? I'll start with a holistic discussion about the three routes of human exposure, which will take us briefly into another biomedical discipline, anatomy and physiology, which deals exclusively with the organ systems within the human body and how they collectively work together at the tissue and cellular levels. So again, this was written for a general audience. Uh, a a major part of science is communicating to the public, and when you're so steeped in a field, whether it's a research lab or in a regulatory discipline, you're not thinking about communicating to the general public. You're not always thinking about that. So again, I showed this to a couple of colleagues before I published this, and one did ask, "Why? Who's this for? Why are you writing this? Who's this for?" So Doug says he asks, "Was animal testing recently banned? No, to my knowledge, it has not been banned, though science is trying to move away from it so that there's less of it. Okay. Let me see if I have that slide handy. Give me a second. Yeah, keep asking questions or commenting there in the chat. These are more fun when it's interactive and when it's not just me talking. I'm hoping I have this slide handy. If not, I'm going to have to wing it. I think it's in here. Yep, there it is. All right. The force is with us this morning. All right. So, routes of exposure. My posts regarding pharmacology, toxicology, and ADME/drug metabolism focused on exposure to chemicals primarily through the oral route. That is ingestion through the mouth and then absorption into the GI tract or the gastrointestinal tract. While we typically think about the ingestion of chemicals through the oral route, the reality is that humans can be exposed to drugs and toxicants through two other routes: the dermal route by way of our skin and the inhalation route by way of our respiratory tracts. The region spanning from our nasal passage down into our lungs, where gas exchange, the atmosphere occurs. Each route has its own unique properties anatomically, which impact the potential absorption of chemicals into the body, where they can exert their therapeutic or toxic effects. Each route receives differing amounts of what's called the cardiac output, or the blood delivered from the heart. On average, the GI tract receives 21%, the skin receives 9%, and the lungs receive 100% of the heart's cardiac output. This makes sense as the function of the lungs is to facilitate gas exchange between our bodies and the Earth's atmosphere. So I'm going to show this on the slide. This is the slide I just pulled up. This is a look or comparison of the three routes of exposure. Okay, so through the oral route, that's basically ingesting a chemical via your mouth that may get absorbed by your GI tract and absorbed into your bloodstream. And your GI tract receives approximately 21% of the cardiac output. There's the inhalation route. It receives 100% of the cardiac output, and there may be something called a portal of entry effect, which I'll talk about in a little bit. And then there's the dermal route, which receives roughly 9% of the cardiac output, of your cardiac output. There's something called the first pass. There's something called first pass metabolism. I'm not going to get into that right now. That would take a while, and I want to get on and off in about an hour. I have some comments here. Let me see what you guys are saying. Doug says I have to look into it. I recently heard a news clip stating new vaccines will no longer test on animals before human trials. I believe I got the animal testing confused. Okay. In gas exchange, I just mentioned gas exchange. Let me show that to you all really quickly. I'm sure you all know what that is, but for the sake of completeness, it's worth showing. Let's see, where is it? So there's the lung, there's hemoglobin, there's the ribbon diagram of hemoglobin. Uh, Everyone, I should have had these queued up before starting, but again, busy week. Okay, ions. The liver. Geez, these things are never around when you need them, and they're usually hiding in plain sight. I see every other cartoon here except those. Okay. Okay, there we go. I need one more. All right, there we go. All right, gas exchange. So your heart, this is the thumbnail for this stream. Your heart and your lungs, they work together. The heart is the pump that pumps the blood all over our bodies. The lungs, our lungs are responsible for a gas exchange. So we inhale atmosphere, we inhale air from the planet. It's oxygen rich. Our uh, that oxygen gets absorbed into our bloodstream. When the blood comes back to the lung, it's carbon dioxide rich and oxygen poor. So there's an exchange that takes place, and this is how we live. This is a basic life process that we can not live without. The rest of our bodies, the rest of the organs in our bodies would suffocate without this simple process. The protein that oxygen is carried in red blood cells, specifically on the protein hemoglobin. Okay. So breathing is a very, very necessary biological process. We have to do it. We can't go without it for long periods of time. I'm sorry. We can't go without it for long periods of time. And so if we're exposed to something in the air, that can be very problematic because we have to breathe. Okay, let me move on. The alveoli and gas exchange. The lungs alveoli are critical for the body's absorption of molecular oxygen into the bloodstream. Once inhaled, the oxygen in the air is very rapidly absorbed into the pulmonary capillaries from the alveolar spaces where it binds to hemoglobin in our blood, while carbon dioxide releases into the alveolar spaces to be exhaled. This exchange of oxygen and carbon dioxide are both very rapid and efficient in healthy lungs, something our bodies do without us even thinking about it. What allows for this is a very efficient of exchange of gases with the environment. Um, I'm sorry. What allows for this very efficient exchange of gases with the environment is a very, very thin 0.5 micron um, three cell layers separating the alveolar spaces from our pulmonary capillaries. These capillaries immediately receive and return blood to the heart for distribution to the body. I'm going to show that picture one more time so you all can see it. This is what that paragraph just described. You have carbon dioxide rich and oxygen poor blood going to the lung. There's an exchange with that airspace, and then the blood going to the rest of the body is oxygen rich and carbon dioxide poor. Okay, I don't want to belabor this. Without the continuous exchange of oxygen and carbon dioxide through our lungs alveoli, our bodies could not function, as oxygen is a necessary substrate for our bodies, many tissues at the cellular and molecular levels. This is important because while other bodily tissues can tolerate um, varying degrees of oxygen deprivation, it is well understood that even short periods of deprivation of the brain can cause irreversible damage, unlike with long periods of food and water deprivation. For this reason alone, maintenance of proper respiratory function is critical. With that, I'll transition into what inhalation toxicology is and why it's important. All right, hazard through inhalation. Inhalation toxicology is the study of the harmful effects of chemicals on living systems through the inhalation route of exposure via breathing, typically as it applies to mammalian species. It's a very important field as respiration is a critical biological process for mammals, as described above. And thus, any toxicant that compromises the body's capacity to exchange oxygen and carbon dioxide with the environment is very, very dangerous. Let's take a quick look at respiration, just so we have a feel for what that oxygen is used for. I should be able to find this one pretty quickly. I hope you all are having a good Saturday thus far. I'll use this one. So the oxygen, what do we use the oxygen for? We as animals, we use the oxygen for respiration. So this is something we learned in undergraduate biology. Respiration and photosynthesis are a coupled reaction and they're opposites. So we as animals, we use the oxygen and the glucose to create something called ATP. That creates carbon dioxide and water. Plants use the carbon dioxide and water to create the sugar on that right side. So in other words, we need the oxygen to perform respiration, and that's how our cells function, and that's how our cells eat. So no oxygen, you die. We die. I die. Before I discuss the types of chemical agents that can cause injury through inhalation exposure, I'll describe the two types of effects that can result from exposure to inhalation toxicants: portal of entry effects and systemic effects. A portal of entry effect is any effect produced in the tissue or organ of first contact with a chemical or toxicant. In this case, it's an effect where a toxicant causes damage starting from the nasal passage down into multiple regions of the lung. There are multiple regions, and cell types in the respiratory tract, each with specific functions, all of which can be uniquely injured. Okay, I think I have some pictures of this. If you guys have questions or comments, please let me know. Okay. Okay. So I'll use this cartoon again. So if you look at that inhalation cartoon in the middle, all of that is your respiratory tract. So there's the nasal cavity, the pharynx, the larynx, the trachea, uh, the right lung, the left lung, and there are and those air passages, those passageways, they branch off into smaller branch passageways down to the alveolus, the alveoli. So there are unique cell types throughout there, and each of those can be damaged by a given chemical or toxicant. Now, going back to let's see who's out there listening. Now, going back to the global health crisis, or global health crisis, I'm not going to say its name, the beer bug. How does the beer bug relate to all this? How does the beer bug relate to the respiratory passage? Anybody? And the beer bug, you guys know I'm referring to C19. How does that relate to the respiratory passage? We have an answer. We don't have an answer. Okay. The lung, the lung was the passageway, the entryway for C19. And I believe it was, I want to say it was the ACE receptor or the ACE inhibitor. Not the ACE inhibitor, the ACE receptor. It's so long since I did content on that. I think this was the I think this is the passageway with which the virus was able to get into our bodies, and that's why the test used a nasal swab, because you'd be able to detect viral particles in your respiratory passage. Well, whether that was the right receptor or not, that's the passageway with which the virus was getting into us, which is why we had to, uh, which is why you had the mask mandates, which may or may not have worked. And once the virus got into your respiratory passage, it could get to the rest of the body pretty easily. That's right. That's right. Doug's on top of it. Yep. Okay. Moving on. In laboratory settings described earlier, some portal of entry effects are instant, while when observing lab animals and manifest as clinical signs, which are visible. Irritation in the respiratory tract can trigger the painful reflexes in bradyapnea in rodents, which are immediate changes in the breathing patterns of the animals through very sensitive nerve processes and receptors in respiratory tissues. Anyone who has worked with worked in a research lab and has opened a bottle of concentrated hydrochloric acid outside of a fume hood appreciates how quickly irritation can occur, as it only takes a few seconds to feel a burning sensation in the nose, followed by coughing, watering eyes, shortness of breath, etc. So, yeah, if you have a bottle of hydrochloric acid next to you and you open it up, it starts vaporizing immediately and it irritates, it gets in your nose and it causes a lot of damage. Okay. Other portal of entry effects. So, a portal of entry effect is the point of contact. So, it's typically going to happen right there. It's going to happen right there in the respiratory passage. So, as soon as that chemical hits the tissue, it's going to do what it's going to do. Okay. Other portal of entry effects are more time dependent and can take hours, days, or weeks to fully set in. Some are reversible, while others are irreversible. Prolonged exposure to some toxicants can cause inflammation in the lungs, leading to pulmonary fibrosis or the formation of pulmonary edema, both of which compromise lung function and eventually can be fatal. Asbestos poisoning causes injury through prolonged activation of the immune system in the lungs, damaging them over time as asbestos particles cannot be removed once inhaled. Okay. I'm not going to get into the asbestos story, but the long and short of it is once those particles get into your lungs, they can't come out, and your immune system tries to clear it, and it basically wreaks havoc on your lungs. Smoking cigarettes is a good example of people willingly injuring their lungs. The paper used to roll cigarettes and the tobacco inside them contain thousands upon thousands of compounds before the cigarette is even ignited. Once lit, those chemicals are combusted. Um, they transform into numerous other chemicals, some of which are referred to as reactive intermediates, which themselves come into contact with cells of the respiratory tract. Years and years of direct cigarette smoke, inhalation can cause irreversible damage leading to diseases like lung cancer. There's also a risk of lung injury from living in industrial areas where there is the potential to inhale combusted compounds and particulates from factory emissions. So, those of you who are sub to the channel, you know that I interviewed a gentleman back in the fall named Floyd Vergara. You can look back in my catalog at that. Floyd was an advocate of biodiesel fuels and clean energies because those would put less contaminants and particulates into the air in urban centers. So, according to his data and his modeling, uh, diesel emissions are decreasing the quality of life of people who live in certain urban communities. Okay. So that's why the biodiesel clean energy, that's part of why the biodiesel clean energy uh, aspect is so important and why people are interested in that. Before moving on, I'll add here that while many inhalation toxicologists consider the lung itself to be the most important part of the respiratory tract, recent science has shown that the nasal passage is also a toxicologically relevant tissue as it relates to inhalation exposure. It contains drug metabolizing enzymes similar to those described in my ADME drug metabolism post. The lungs do as well. Some chemicals can thus damage these regions if inhaled for prolonged periods of time. I'm going to show you guys something That picture right there, I have that model sitting in a closet back there in my home. That is the human nasal passage. This is a picture of the rodent nasal passage, because oftentimes the test model, as I'll describe towards the end of this, the test model for these experiments right now are rodents. So we are moving towards more in vitro and in silico methods. So more cell-based and more computer model-based testing. But this is what the rodent nasal passage looks like versus ours. Okay. That is I want to say a longitudinal illustration of it. Okay, here is another image of it. It's divided into those regions: the nasal turbinates, the nasal vestibule, the maxilloturbinates, the ethmoturbinates, and the olfactory recess. And this is the cross section of that nasal passage. These are cross sections of those regions. So if you cut the rodent nasal passage up, you'll see images like that. And the significance of that is that when you're conducting these experiments, which I'll discuss at the end of the stream, you have to, when you're comparing the human nasal passage to the rodent nasal passage, they're not the same. So you have to scientifically account for that difference in their design. Theirs is more convoluted. It's designed to filter out certain things. Ours is not as convoluted. Shonda says, "Good morning" to myself uh, and to the chat. Vaping is being associated with lung-related inhalation injury too. For eval, what is Shonda? What does that mean? Evali? What does that mean? Is that a product? I'm not sure. I think I tried the vaping once or twice, so I don't know enough about it. But yeah, that does create well, it creates a vapor as opposed to a suspension with particulate in it, like cigarette smoke. But if it's being implicated with lung cancer, then that's bad for the industry. UFO Kamikaze says initial reports of C-19 victims describe some having lung damage similar to that of first responders from the World Trade Center. Those initial reports stopped being aired and mysteriously disappeared mostly. Yep, not a surprise. Not a surprise. Okay, systemic effects. Systemic effects refer to injury slash toxicity in other parts of the body beyond the respiratory tract. So, in other words, if you inhale something, in some instances, it can go through the respiratory tract and then end up in your general circulation. Okay, which is dangerous as well, and I'm going to talk about why. If a chemical slash toxicant can efficiently pass through the lungs alveoli, as described later, it can enter the bloodstream and into the body's general circulation. From there, it can damage other organs, as discussed in my toxicology post. Medicinally, some therapeutics, such as anesthetics for surgeries, are actually administered this way. Halothane is an example. So if any of you have had surgery, like me, sometimes they can give you an anesthetic and put you to sleep by inhaling a drug. Or if you're post-op and you're having a fever, they can give you an aerosol to calm your body down. I'm going to talk about what an aerosol is shortly. Shonda says EVALI is the acronym for e-cigarette vaping associated lung injury. Shonda, thank you. We're all learning here. Now I know what that means. Please smash that like button if you haven't. Two classic systemic inhalation toxicants are carbon monoxide and hydrogen cyanide, which I've hyperlinked in case you're curious to learn more about how they work. While carbon monoxide poisoning has been associated with um, accidental deaths from tailpipe emissions in garages, hydrogen cyanide is a known potential chemical weapon, which is particularly dangerous in enclosed spaces such as subway stations, something our intelligence agencies are very, very aware of. So if you all watched the what do you call it? The Equalizer with Denzel, Ryan Coogler, the Equalizer, if you watch the first one, when Denzel was tracking down the bad guys one by one in the first movie, you remember he took the crooked cop, handcuffed him to the steering wheel, and then put the tailpipe. I think he put the tailpipe inside the car, and he kept revving up the engine to eventually make the guy cave to tell him who the bad guys were. And you notice the guy kept coughing and coughing and coughing. That's because carbon monoxide was coming out of the tailpipe, and carbon monoxide it binds our hemoglobin very, very efficiently, which is why it's so dangerous. So in that movie, Denzel was uh, basically torturing the guy to get him to give up the other bad guys. But that's why it's very dangerous to have a car engine running in a closed space, because the carbon monoxide is coming out very, very quickly, and you can't smell it, and it will suffocate you and kill you. The same thing is true if you're in a home, and I think if there's a gas leak of some sort, the carbon monoxide will kill you pretty quickly because you can't smell it, and it binds your hemoglobin very, very efficiently. All right. And the hydrogen cyanide, that's there's a concern for that in subway systems because it's a closed space. And in theory, if someone throws a jar of it on the tracks and a train is coming, all the air is going to push towards the people on the platform and kill everybody. So that's systemic toxicity through the inhalation route. Please hit that like button if you haven't. If you want to make a donation to the channel, this information is here. You're under no pressure to do so. Charlie says, yeah, seen the movie. Yeah, I think we've all seen that. Okay. These are just a few types of, these are just a few examples of toxicity through the inhalation route of exposure. There are many other chemicals and substances that can cause injury, and in some cases, therapeutic benefit through the inhalation route of exposure. Many industries and groups highly consider inhalation toxicology. They include, I'm going to get to that in a second. What's another, for those of you out there, what's a therapeutic benefit, or how is the inhalation route used therapeutically? Let me ask you that. Does anyone know? How is the inhalation route used to treat a disease? There's a very common condition a lot of kids have had, particularly in inner city communities, and this condition is treated using the inhalation route. Who knows what it is? Yep, UFO kamikaze got it right. Yep, it's asthma. So kids who have asthma, they carry around this nebulizer. There's a nebulizer. I'm not sure if that's the right word for it. This inhaler, we'll call it an inhaler, and it creates an aerosol, and they inhale the albuterol, and it causes their respiratory tract to relax. Hey, Aquateki, thank you for stopping through. We are almost done, I promise. So inhalation toxicology, it touches all of these industries. I'm not going to read this for the sake of time, but the chemical industry, the tobacco industry, um, nanoparticles and nanomaterials, national defense, the pharmaceutical industry and medical devices, and public health. And this picture here in the DMV, uh, before the global health crisis, when you would get on the metro in the morning, uh, they would hand out free copies of the Express. And one morning there was a this picture was there of I guess there was a gas attack in Syria. So when you think of something like nerve gas, sarin gas, all these things you hear about happening in the Middle Eastern countries, those are inhaled toxicants. And this little kid here, unfortunately, got an inhalation exposure to some of those gases. Okay, I'm going to wrap up with the research, and I won't hold you too much longer, I promise. Having introduced the field in terms of background and context, I'll now discuss some of its experimental and technical aspects using visuals provided by CH Technologies, a leading company in the manufacture of inhalation toxicology exposure systems. Inhalation toxicologists and scientists not only need an understanding of the biology of injury to the respiratory tract via inhalation exposure, but they also need an understanding of how to properly create the experimental conditions to test for inhalation toxicity. It's relatively straightforward to feed a test specimen the chemical of interest in food or water, or to apply it via the skin, but how do you administer it for inhalation exposure? the answer is that the chemical must be administered as a gas, an aerosol, a dust, or even a cigarette smoke suspension in some instances. This involves some knowledge of chemistry, physics, as well as mathematics and statistics. A key aspect of any toxicological field is proving the concentration slash dose tested and properly correlating it with the effects observed. Scientists must thus be able to verify their test atmospheres, and there are numerous analytical chemical methods for doing so. So you basically, when you're conducting these experiments with animals, you have to create a test atmosphere where you're introducing the chemical as a gas, an aerosol, or a vapor, or a cigarette smoke suspension, or a suspension of nanoparticles. I want to show you what that is shortly. Some chemicals readily exist in the gas phase. That is, they have what is referred to as a high vapor pressure, and they are volatile. Some are liquids while others are solids. Mothballs are an example of a volatile substance, a solid which sublimes and converts directly into a vapor. They give off a unique odor, most of us know from our grandparents' closets, and are comprised of the chemical naphthalene. which itself has a high vapor pressure. Other chemicals have a low vapor pressure and are considered non-volatile and must form aerosols to be inhaled. Think of a mist from a spray bottle. Dust suspensions can be generated as well for experiments. In some instances, generating inhalable suspensions are not feasible depending on the properties of the test material of interest. So, gases, aerosols, and dusts, I think you all intuitively know what a gas is. That's an image of a gas. I'm not sure how that's if that's infrared or what, but you have gases. There are also aerosols. Oh, I'm in the wrong folder. Typically, when you spray something out of a bottle, that's an aerosol. So you're thinking about droplets of some sort, and there are a lot of considerations for those as well. And then in some instances, dusts and particulates. You may want to give the animals dusts or particulates depending upon what the product is. So gases, aerosols, dusts, and in some instances, cigarette smoke extracts. And cigarette smoke contains a bunch of different things. UFO Kamikaze, this is a very compelling comment. He says, what may initially seem far fetched sometimes becomes less ridiculous as more information is revealed. Okay, we are almost at the end, folks. So, test models and species. While the test species for inhalation toxicology studies vary, the species of choice is typically rodents, rats, and mice. In some instances, guinea pigs and primates are used. Each of these species possess the same organs that humans possess for the most part and are thus useful models for human exposure. Scientists must be well trained in both caring for the test animals and also operating the highly specialized equipment used in these studies, which I'll cover next. Okay, so the test system, testing a drugs slash chemicals efficacy slash toxicity through inhalation exposure requires use of an exposure chamber where an inhalable atmosphere of the test article is generated for inhalation exposure by the test subjects. The accompanying picture shows a single level chamber with the accessory equipment used for measuring the chamber's inner atmosphere using some of its exposure ports. Click on the images to Click on the image to enlarge it using the accessory equipment. The concentration of the test material in the chamber can be monitored by scientists running the experiment, in addition to other important measurements, including oxygen, carbon dioxide, temperature, and humidity, to name a few. Let's see if the enlarge still works. Yep. Boom. So that is an exposure chamber. Okay. That cylindrical structure there with the ports on the side, that is an exposure chamber. What you do is the animals, I'll show you, well, I'll partially show you. They sit in these restraint tubes and you plug them into the chamber and they inhale the atmosphere. Now that's one stack. But what you can do is if you want more animals, you can stack the chambers for multiple levels. Let me see if I have the picture here. Okay, where is it? Okay, this this is a picture of two chambers stacked one on top of the other. So if you look at that, yeah. So if you look at that lower tier, I believe this is the one I actually used when I was doing the experiments. I think that has 20, that has 12 ports on the bottom. And so if you add 12 and 12, you get 24. So you could test a total of 24 animals at one time, which I was able to do during my postdoctoral studies. So that's an exposure chamber. But what you want to do is you want to make sure you're monitoring when you have the animals plugged in there. You want to make sure you're monitoring the oxygen and the carbon dioxide and the humidity because they're breathing. And if the oxygen goes too low, you could kill all the animals. All right. To generate the chamber's test atmosphere, most modern systems utilize an air pressure pump to create an inflow and into the exposure chamber and a vacuum pump to create an outflow from the chamber, together creating a consistent supply of oxygen and removal of carbon dioxide for the test subjects. The accompanying diagram shows a complete inhalation exposure system designed to expose test subjects to aerosols. Click the image to enlarge it, whether gases, aerosols, or dust are generated. A supply line for the test article is ligated into the air supply line, feeding the exposure chamber, allowing for the control of the concentration within the chamber by the scientist, something that must be actively monitored throughout the experiment. So that is an inhalation system right there. So you have your exposure chamber, that's your apartment building like thing with the that's those are the chambers stacked on top of each other. And then over here towards the lower left, those are your controlling apparatuses. I'm not going to go into all that. This link is below in the description box in case you guys want to take a closer look. But this is an inhalation exposure system. So there's a lot that goes into using this type of thing. Okay, I'm going to show you guys a few more pictures, and we're just about at the end here. I don't think my postdoctoral advisor will mind me sharing these with you all. I mean, this was over 14 years ago. So this is the exposure chamber. This was in a hood, right? This is the actual control panel right there. So you can see the exposure chamber back there in the hood, and this entire black panel is how we controlled what went into that chamber and what came out of that chamber. And Yeah, it was a very, very complicated device. If you look at that white cooler right there, that contained the pump and the vacuum, which made quite a bit of noise. I see a comment here. Um, UFO Kamikaze says blood oxygen monitors were initially used to diagnose suspected C-19 patients by paramedics. Yep, I imagine so. I imagine so. Let's see. So inhalation studies can use whole body chambers where the animal's whole body is exposed, or nose only or head only chambers, which in some instances have become preferred methods due to their increased specificity to the respiratory tract. A potential drawback of using whole body chambers is that test subjects, usually rodents, in the process of grooming themselves, can orally ingest the test material by licking their coats. Dead space within the whole body chambers is also a drawback. The accompanying picture shows rodents in a restraint tube during exposure. An important key to properly running inhalation exposure experiments is making sure that the animals are adequately acclimated to tubes and are comfortably in them for extended periods of time before the experiment. So, that's those are IKEA mice, and CH Technologies was nice enough to donate that restraint tube for me. And yesterday, I was thinking, one of the hard parts about the biological sciences when you're testing chemicals is sometimes you are testing using live specimens like rodents, like lab mice or lab rats. And when I was doing these, I had to get the mice used to being in those little small tubes, and it was uncomfortable for them initially. They get used to it, but um, it's important to treat the animals as humanely as possible, and so that there's so that they undergo the least amount of suffering and discomfort for their sake, and it can also impact your data. But that's something I had to get used to. Was that there were two stretches in my career or in my training where I worked with animals, and that's something you have to get used to. Is ultimately sacrificing the animal and also potentially putting it through something that may be stressful for it. And these experiments, putting them in those restraint tubes for long periods of time, that was stressful for them. So I kind of felt something yesterday as I was thinking about this and thinking back to this particular work. So we're almost done. The accompanying photograph shows a nose only, uh, a nose only inhalation exposure chamber with all of its exposure ports occupied by the restraint tubes for rodent species. Uh, Click on the image to enlarge it. The picture further shows how the number of animals exposed can be increased by stacking multiple chamber levels and increasing the total number of exposure ports. So I'm going to enlarge this so you guys can see it. So imagine you have 36 animals plugged in there, actively inhaling whatever it is you're giving them. So that's how these experiments are conducted. Um, and the significance of nose only is that the cone, it literally comes down like this. So when you put the animal in, when you put the rodent in, their nose is literally at the front of the cone. So it's inhaling whatever is in there. But again, you just have to make sure they are as comfortable as possible when they're in there. I couldn't imagine being restrained for that long, for hours in a space that's small. But animals are very resilient, maybe more so than humans. I mean, even though we are animals technically, but it's something to see. Depending on the questions being asked in that particular experiment, exposures can range from hours to days to weeks to months and years. During and afterwards, any number of toxic therapeutic biological responses can be measured, including changes in clinical signs, body weights, blood chemistry, clinical chemical parameters, and changes in organ weights and tissue microstructure or histopathology. Again, collectively, these are a very technical set of experiments to run and which require a very specific and unique skill set. So when I say the animals can be in there for hours to days to weeks to months to years, that is not continuous. You do let them out to take breaks, or if you're conducting an experiment over a month, you expose them for a certain amount of time every day, and then you let them out, and you come back and you do it again the next day, and then you let them out and back into their cages, and you do it the next day and the next day and the next day. So they're not just in there all day, all night, all year. They would die that way. So the training, this is arguably the most important part of this. How can students get trained in inhalation toxicology? Beyond high school, students can major in biology, chemistry, or any of the biomedical sciences as undergraduates, where they can start receiving lab training if there are researchers at that particular university or one close by. Further training can be obtained at the master's or PhD levels, similar to pharmacologists, toxicologists, and drug metabolism scientists. Inhalation toxicologists generally receive their training at major research universities. So you're typically going to go to the University of Michigan, Stanford, MIT. There's an actual center that specializes in inhalation toxicology called the Lovelace Center. I believe that's in New Mexico, but you have to go to a um, a major research university to get this training. I want to thank Darnell Smith for the generous Cash App. Wow, thank you for that, sir. That was thank you. That I really appreciate that. That means that looks like you've gotten some out of this. Thank you. UFO Kamikaze says, anyone remember the massive bird deaths that were happening? Makes you wonder if flight travel bans were put in place to stop a virus spreading or to keep passengers away from point of origin. UFO Kamikaze, I don't remember that. Were they restricting travel to certain parts of the globe? I don't remember that. Okay. As a subdiscipline of toxicology, scientists looking to receive training in inhalation toxicology can have varying backgrounds in terms of degrees conferred. If an individual doesn't initially train in an inhalation toxicology lab, they can work in these labs as postdoctoral scientists or fellows with any of the biomedical degrees, and even with medical or veterinary degrees. When I gained my training in inhalation toxicology, my PhD was actually in pharmacology. Okay. So it's the skill set, regardless of when you're in the sciences, regardless of your degree title, it's your skill set that's the critical thing. Depending on the degree level earned and where the scientist is employed, inhalation toxicologists can earn starting salaries of $60,000 to $70,000 and above. One of the themes of my posts in this series is there is a tremendous amount of flexibility and overlap in the biomedical sciences. Upon receiving training in inhalation toxicology, scientists must then determine which sector they want to pursue: academia, the private or public sectors, or non traditional careers. Scientists with this background also have the flexibility to combine their knowledge sets with other disciplines to go into a wide variety of areas in pharmaceutical companies and biotechnology companies, chemical companies, consulting companies, patent law companies, and even starting their own companies and contract labs. Okay, this is the ending. We're finally here at the ending. Let's see. Doug says Don Cheeto issued a travel ban from China and Italy, but then he was called xenophobic. Yeah, I don't know if we should get into all that. Yeah, I don't know if we should get into all that. Anyway, UFO Kamikaze says point of origin equals atmosphere and upper atmosphere. Okay. Doug says he stopped a few cruise ships out at sea for 2 weeks. I mean, based upon what was happening at the time, it was the smart thing to do. Okay. This is usually where engineering cannabis drops in. Toxicology for the 21st century. It's worth reiterating something from my toxicology blog post, and that is that there's an effort currently underway called Tox 21 or Toxicology for the 21st century. One of the goals of Tox 21 is to minimize animal usage. Currently, there are efforts to develop methods to test for inhalation toxicity using in vitro models and cell culture preparations, simulating animal tissues. Students interested in this field will position themselves well by learning about some of these advances that are on the horizon. So, yeah, so I'll read this last paragraph. Thank you for taking the time to read this post, and I hope I was able to shed some light onto what inhalation toxicology is as a field. Similar to the other disciplines I've discussed, inhalation toxicologists have their own professional societies and meetings. While the Society of Toxicology has subsections on inhalation toxicology, the field has two of its own professional societies and meetings. They are the American Thoracic Society and the American Heart Association, as the heart is a major organ affected by the inhalation of toxins. All right, this is below in the description box, everyone. I'm going to go back because I did create a slide on this. I'm going to wrap this up. But I wanted to show it since I went through the trouble of making it. And I think I forgot to show something else as well. Yeah. So when you're testing animals in these experiments, these are things that, and I read this in the post, it's summarized there, but it's good to see them in bullets too. These are things you typically measure: clinical signs, changes in body weight, blood chemistry measurements, organ weight changes, macroscopic changes, and microscopic organ changes. So macroscopic organ changes and microscopic organ changes. So those last two, what is a macroscopic change? So we'll take a look at this model here. If you notice in the model, the lung is it's pink. Well, it's got a reddish color. That's because there are blood vessels innervating it and moving all throughout it. The heart is pumping blood into the lungs, and blood is going back to the heart and going to the rest of the body. So if there's some sort of damage to the lung, you might see the color altered. So it might go from a pink to a deep red. A deep red would indicate that there's some sort of massive damage there taking place inside of it. So that's a macroscopic change or alteration. A microscopic change would occur at the cellular level. So you'd have to sacrifice the animal, remove the lung, fix the tissue, and take microscopic sections. So you can look at it under a microscope and look at the cells to see if there were alterations in morphology. So there's macroscopic, which is kind of high level, and then there's microscopic or histopathological alterations. That's something a pathologist would do. And then UFO Kamikaze says, remember seeing gray dust on this vehicle in the morning, similar to pollen or something shortly before and early on during the World Health Crisis. Others online mentioning seeing things too. Well, well, think of back to 9/11. Was it 9/11 or was it in the early 2000s? I don't know if it was the Unabomber. There was a concern for someone mailing white powder, mailing white powder to politicians or whomever else. That was anthrax. So that would also result. The concern was that would also result in an inhalation exposure. And once that anthrax gets inside of you, you're going to get pretty sick. Yeah. So I'm going to go over this one last thing. I forgot to mention it during the rest of this talk, and then I'm going to wrap this up because I want to try to get out to the gym and come back because I have another interview to do. Vapor pressure. I mentioned vapor pressure and volatility. So vapor pressure, in this instance, we're thinking about gases, right? So vapor pressure is the equilibrium. Vapor pressure is the equilibrium pressure of a vapor above its liquid or solid state. So I'm going to read these bullets. At equilibrium, the evaporation rate equals the condensation rate. Molecules in vapor phase collide with the walls and lid of the container, causing pressure. Evaporation and condensation occur at the liquid surface, and increasing the temperature increases the rate of evaporation and increases vapor pressures. So some chemicals have a high vapor pressure, which means they readily go into the gas phase. And that's different from, and so those chemicals are considered to be volatile. Whenever you hear that a chemical or a substance whenever you hear that a chemical or a substance has a high vapor pressure or it's volatile, what that means is that that chemical, based upon its properties, has a very low boiling point and it's going to readily go into the gas phase for inhalation. If it's non-volatile, it's not going to go readily into the gas phase and it has to form an aerosol in order to be inhaled. So those are two principles that I talked about during this post that I didn't get into the weeds about, but that are very, very important. Is it volatile? Does it have a high vapor pressure? Or is it non-volatile? Yeah, Aquatech, you can think about that as well. But what and I'm going to end with this. For the beer bug, the reason they wanted us to use these, and here in the DMV, some people are still wearing these and wearing these on the metro. The beer bug, the concern was that the beer bug would spread through aerosol formation and transmitted by aerosol formation. And so the masks, in theory, would decrease the likelihood of aerosols being transmitted from one person to the next. Aquateki, thank you for the generous Cash App. I appreciate that. Appreciate the donations I've gotten over here. This isn't the most saucy stuff, so I appreciate you guys hanging out. US UFO Kamikaze says Sicko Mode, Travis Scott and Drake music video intro released just prior to the World Health Crisis, also worth checking out. Just Ashar says probably will be wearing them to 2025. Who will? Who will? It looks like it it is it's considered over. I know that for the government, at least here in the DMV, they want to start to call everybody back to the office, and I think you're seeing that all over the place. The desire to get workers to come back to the office. So I don't know. So I don't know if people will be wearing these for a while. Oh, she says older people. Okay, older people. Okay. Kareen of House of Trades, thank you for stopping through. I'm about to wrap this up. I need to go get some exercise. I have something else to do this afternoon. So everybody, thank you for being here. This this was a complex topic with some very complex material. And at some point, maybe I'll come back and actually show a paper and some data so you all could see what an actual paper and data looks like with the interpretation and with the statistics and with everything else. But I thought it was important to present the science, the discipline, and the concepts first. And this is a very unique skill set. Not everybody has this, and not everybody has this training. And I think it's important, particularly for individuals of my background and your background, the a lot of communities don't know what this is, and that's why when I would go do career days here in the DMV, I would get, "You're the first Black scientist I've ever met," and that's why even amongst the federal workforce, other government workers would say, "Wow, you're the first scientist I've ever met. I've never heard of inhalation toxicology. I've never heard of pharmacology. I've never heard of uh, drug metabolism." Because in our communities, people just don't know. And we just, I think we're just concerned about other things. There aren't a lot of us, so there aren't a lot of examples. And I think we're just concerned about other things. Master Teacher, I see you stopped through. Thank you for stopping through. I see you said you catch the replay. Okay. You have a chemistry background, so I think you'll be able to follow all of this pretty easily. But yeah, I appreciate that you stopping through, and I hope you do take a look at this. This is how the inhalation was the route of exposure of concern for the beer bug. So, and this has a lot of applications in a lot of different areas. So, let's see. So I'm gonna wrap this up. So everyone, if you're new, those of you who have been here, you know about my newsletter. If you're new, please consider joining. There's a 2-paragraph greeting at the bottom. You just click that subscribe button, and it takes you here. And you can just enter in your email address and sign up. It's that easy. And I promise I will protect your information. Let's see. I'm gonna show a little extra really quickly. before I go, it looks like Elon Musk just made another advancement with his rockets. Fair use, fair use. He's been this image has been making the rounds. That rocket ship launching off. I'll let it go one more time. Fair use, fair use. YouTube, I don't know if this constitutes a violation of any kind, but I thought that was kind of cool. It says Starship, he tweeted, Starship liftoff in slow motion. Okay. All right, everyone, I'm going to wrap this up. Thank you to those of you who hung out and listened to me talk about this for the last hour and a half and commented and asked questions and gave your thoughts and your feedback. If you're new, please like, share, and subscribe, and especially share, especially for individuals who have never heard of this before, especially young people who've never heard of this before. So with that, I'm going to wrap up. Look out for the next one. Everyone stay safe out there. Enjoy the rest of your weekend. As always, remember that your attitude determines your altitude. Always try to do your best. Take care, and I'll talk to you the next time. And if you have questions or comments after watching the playback, please let me know what you think in the comment section below, or ask me anything in the comment section below. All right, take care. Bye-bye.